Hello, Bio students. Today we are going to continue with our unit on classification, and we are going to focus on the lesson on bacteria. So we are going to introduce prokaryotes, which we know are bacteria. So what is microbiology? So microbiology is a field of science that focuses on studying microscopic organisms. And remember that microscopic organisms, micro means very, very small. So these are organisms that we cannot see with our eyes. So we need to look at these organisms under the microscope. So some of these microscopic organisms that microbiologists study are prokaryotes like bacteria, but it could also be viruses, and it can also be tiny little eukaryotes such as protists and fungi. So who is known as the father of microbiology? Well, considering that we wouldn't have the field of microbiology without a microscope, then we say that Anton van Leeuwenhoek, who was the first one to use the microscope, and we saw his discovery of the microscope and the simple microscope that he used in previous units, we give him the credit, right? We call him the father of microbiology. He was the first to identify and describe what he called at the moment as animalcules, or unicellular organisms, and this was in the mid-1600s. So we consider bacteria, some of them at least, to be pathogens. Remember that there is also good bacteria, and this is part of your natural bacteria that you find in your intestines, for example, to help you digest food. But there may be some bacteria that are pathogens or pathogenic. And these are the types of bacteria that cause a disease or make you ill. So we refer to these pathogens or these types of bacteria as germs or microbes. Now, the reason why they make us sick is because they can secrete some forms of poisons or toxins, and they can disrupt our normal metabolic activities, and they can disrupt our homeostasis. So a long time ago, and we're going to get into this theory in a little bit more detail, the germ theory of disease was proposed. And the scientists who proposed this said that microorganisms, that tiny little organisms that we couldn't see with our eyes, were the cause of many diseases. Now, believe it or not, this was highly controversial when it was first proposed. Can you think as to why that is? I'll let you think about that for a sec. So once this germ theory of disease had enough evidence and it was confirmed by a couple of scientists, it led to very important innovations as using antibiotics through the discovery of antibiotics and applying those antibiotics to kill these pathogens or these bacteria. And also very important hygienic practices. So it might sound weird for you to hear that doctors in early 1800s didn't use gloves to go into surgery, and that sometimes they didn't even wash their hands to go into surgery. And that's because we didn't really understand about these microorganisms and how these microorganisms can get you sick. So now that you've had a little bit of a chance to think about why this theory was controversial when it was first proposed, think of the context of the time period, right? This was in 1840. So in 1840, for someone to propose that there were these microorganisms hanging out in a surgeon's hand, and it was the fault of the surgeon that sometimes patients would die after a surgery because they didn't wash their hands. This was controversial, right? So a lot of surgeons and doctors of the time felt that they were being blamed for deaths of their patients, right? Of course, it was unknowingly that this was happening, but indeed it was happening. Another thing was that around this time in the 1840s, if you were a surgeon, it's very likely that you were a well-educated person, a well-educated man. And as a surgeon, you were a gentleman. And gentlemen did not really carry diseases around those time periods, right? It was seen as a no-no. So to claim that a surgeon who's a gentleman is carrying microorganisms that cause the disease was not seen with good eyes at the time. But of course, it was true. And it was confirmed in the 1870s and through this time period of the 1870s and 1880s with the series of elegant experiments. Here is Louis Pasteur's swan neck flask experiment, where he was able, with evidence, to collect bacteria in the swan of a neck of this flask. And we're not going to go into the whole experiment right now. But his experiments proved that microorganisms actually exist, and that microorganisms that are in the air can sometimes just land, right, in a person's hand, in a person's clothes, inside a flask if it's left open. And many of these microorganisms, it could be bacteria, it could be viruses, could indeed make you sick. So here we have some examples of disease-causing pathogens like anthrax, rabies, the plague, cholera, tuberculosis. So then Robert Koch proposed that through the germ theory of the disease and Koch's postulates that it was time for people in the medical profession to follow certain hygienic practices, right? Such as washing their hands, such as wearing gloves, such as sterilizing surgical tools after a surgery. Believe it or not, right? It might sound strange to hear that that wasn't done before, but indeed it wasn't. And to wear a face mask to prevent these pathogens from being transmitted from the doctor performing surgery to its patient. All right, so here we have a slide that's quickly comparing the eukaryotic and the prokaryotic cells, and we know that no means that these prokaryotic cells or these bacterial cells do not have a nucleus. They do have a nucleoid region, and this is just a region where the DNA of the bacteria is found, right? So we are going to focus on these prokaryotic bacterial cells without a nucleus. So we remember from previous lessons that there are two prokaryotic domains. We have the prokaryotic domain of bacteria, and this is the common bacteria that you find everywhere, and the prokaryotic domain of archaea. Remember that this is the bacteria that lives in extreme environments, and we know that they vary, right, because they vary in the type of ribosomes that they contain, and they also vary in the cell wall composition. The third domain, as we studied in previous lessons, is the eukarya domain, which includes the protist fungi, plants, and animals. So today, since we're studying bacteria, we're going to focus on the prokaryotic domains. So there is a basic structure for bacteria that you should know. So now this doesn't mean that every single bacteria has all of these structures. There are some bacteria that do not have flagella, but these are common bacterial structure. So bacteria have inside DNA in this nucleoid region. Bacteria also have a cell membrane. It's not drawn here, but there is a cell membrane as well that surrounds that nucleoid region. As you can see here, there are ribosomes inside the bacteria. Following the cell membrane, we have the cell wall, and then in the most exterior part, we have the capsule. And then, like I was saying before, not all bacteria have flagella. Some do, some don't, and this is used for movement. And some bacteria have pili, which are these appendages, these hair-like appendages that can sometimes be used for motility, but for most of the time they're used for communication from one bacteria to the other. All right, so we're going to be looking at major characteristics of bacteria. We're going to be looking at the cell shape, the cell wall structure, and the motility or movement. Oops. All right, so there are three basic shapes of bacteria that you should recognize. We have the cocci shape, which is the round spherical shape. There's the bacilli shape, which is the rod shape. And then there's the spirilla shape, which is the spiral shape of the bacteria, or corkscrew form type of bacteria, or filamentous type of bacteria. Okay. Now, there's common cell groups. That means that sometimes these bacteria will form a chain. These type of bacteria are called streptococcus bacteria. This, for example, the bacteria that gives you strep throat. And sometimes bacteria form these clusters, and usually those bacteria are called staph bacteria. You might have heard of staphylococcus bacteria. Almost all prokaryotes have a cell wall outside the cell membrane, and the bacterial cell wall, if you're part of the eubacteria domain, your cell walls are made up of this polymer of sugar, which is called peptidoglycan. Again, archaea do not have this type of peptidoglycan. All prokaryotes have a plasma cell membrane. Most have a supportive cell wall that varies in thickness. And some sometimes have an additional cell membrane, and most of them have a sticky protective capsule outside the cell wall that helps the bacteria against attack from our immune system and also attack from antibiotics. The bacteria can sometimes move depending on the type of bacteria, and they can move by one or more flagella. And remember that flagella is the tail. There are different types of flagella arrangements. They have different structures and patterns. So just understand that the flagella can be one or multiple tails on a bacteria. There's other forms of motility. We have these small little hair-like appendages called pili that can vary in size, and even the bacteria can use it for motility, but most of the time the pili is used so that one bacteria can communicate with another bacteria. Bacteria, they like to form groups of cells called colonies when they're grown in the lab, and you can actually spot these colonies. It's just an accumulation of groups of bacteria, and all of these bacteria are clones of each other because they are reproducing asexually. So you can see here all these little dots that vary in size are the different bacterial colonies. So how do the bacteria obtain energy? So remember that the word autotroph means that an organism can make their own food. So we learned that plants are autotrophs because they can photosynthesize their food and they use sunlight to make their glucose. Now bacteria can also do that, although there are different types of autotrophs. We have phototrophic autotrophs. That means that the bacteria get their energy from sunlight. And then we have chemotrophic, oops, forgot the H here, chemotrophic autotrophs, and this is bacteria that obtain their energy from inorganic molecules, so not the sun, but inorganic molecules like hydrogen sulfide, nitrite, sulfur, and iron. So here's an example of a photosynthetic bacteria, a bacteria that can undergo photosynthesis. They live in moist soils and in water, and since they're photosynthetic, of course, they need light. Bacteria can divide very quickly. Here on the right, we see a single E. coli bacteria that is dividing, so it's making copies of itself. So we know that bacteria reproduce quickly via asexual reproduction. And the example that you need to know is binary fission. So let me quickly move to the next slide here. And here's a picture of binary fission. So this just means that you start off with one bacteria. This bacteria replicates its chromosomes or DNA, and then you end up with two identical copies or two identical clones. Bacteria can also reproduce asexually through budding. So here we see a budding bacteria, and this is where the cell's genetic information is also duplicated, creating an identical copy or clone of the original cell. But now you have, it stems off or it pinches off from a tiny little bud. It's a little different than binary fission. And we know that bacteria, there's no male or female bacteria, right? So we know that bacteria cannot reproduce sexually. But we do know that bacteria can change its DNA by acquiring new DNA from another bacteria. So this is what we call exchanging of DNA. So there's three ways that a bacteria can exchange its DNA with another bacteria. One is conjugation. That means DNA passes through an extension of the surface of the bacteria, and this is usually through an elongated pili. Or transformation. This is where bacteria picks up pieces of DNA from the environment. Or transduction, where viruses that infect bacteria carry DNA from one bacteria to another. So a bacterial DNA can be exchanged with another type of bacterial DNA, and that could lead to differences in the type of DNA within the bacteria. So what are antibiotics? So we see these pictures here from a while ago where penicillin was, of course, branded as the best drug of the time, right? Because it could cure gonorrhea and it could help bring wounded soldiers home. So penicillin is an example of an antibiotic, and an antibiotic is a medicine that is used to fight bacterial infection. So we don't really think of many people dying nowadays because of bacterial infections. Which was something that would happen a lot before the discovery of antibiotics. Oops. So who discovered antibiotics? So it was actually through a moment of serendipity that Alexander Fleming discovered penicillin. So he was a Scottish biologist, a pharmacologist in botany, and he discovered the antibiotic substance now known as penicillin from a mold that was growing in one of his petri dishes that he left in his lab. So what happened was that when you leave petri dishes with a certain medium, bacteria can grow. So here you see normal bacterial colonies growing on the petri dish, right? But then he noticed that there was something here that was killing the bacteria. And there was this area of inhibition here in this area that would inhibit the bacteria from coming into this area. And then when he studied what this thing here growing was, he found this structure that looked like a branched little tree. And he realized that this was penicillium, or a penicillium colony, which is a fungi. So, of course, he understood that this fungi was killing bacteria. So, with the help of Howard Florey, and Ernst Boris Chain, who also won the Nobel Prize along with Fleming, Florey and Chain were able to take that penicillin, which is a fungi, and they were able to convert it into a medicine, which we now call penicillin, that can be injected. You can also take it in the form of a pill. And this medicine or antibiotic is going to kill the bacteria. So it's a good thing, right, that we have these antibiotics, but it's also very important to make sure that we use antibiotics properly. They fight bacterial infections. They kill bacteria, but they don't kill viruses. So if you have a viral infection, you cannot take an antibiotic. And we know that bacteria, since they possess genetic variations, sometimes they learn how to fight these antibiotics. And this is what we called antibiotic resistance. And we know that something is causing this antibiotic resistance, and we know that it's natural selection, right? That sometimes there are these bacteria that have a way to fight off this antibiotic, and they're the ones that survive. So since they survive, they get to reproduce, and then they become more prominent in an environment. So if we ever get sick because of an antibiotic-resistant bacteria, and then somebody tries to give us an antibiotic, then that antibiotic is not going to work, right? So we have to be very careful about taking antibiotics. So we need to make sure that we only take antibiotics under doctor care, antibiotics that have been prescribed, and that we use them properly. We need to make sure that we finish the prescribed dosage of antibiotics so that we kill off all bacteria and we don't allow the bacteria that remains a chance to decipher the antibiotic and become antibiotic resistant. We also need to support organic farming because we know that that is a way. We know that many of these animals in these industrial farms, they are giving antibiotics, and the antibiotics is found, for example, in their milk, and that could increase antibiotic resistance in the future. So that's it for lesson 4. If you have any questions, please go back, and I hope you enjoyed it.